Good morning to all of you. Uh, we have completed the methods of sterilization, which is related with plant, cell, and tissue culture. Different methods we have studied till now. Now, the next part is nutrient medium. It is also important part of biotechnology because without a nutrient medium without a medium or without a culture medium, the process of biotechnology or its related techniques cannot be initiated. So, as you are seeing in this video, nutrient medium topic, I will discuss some of the nutrients and their role. So, how you will define a nutrient medium? A nutrient medium, or you can say, or culture medium, provides combination of nutrients and hormones. So, the another name of nutrient medium is culture medium also, and it provides combination of nutrients and hormones. There will be group of hormones and nutrients. What is the function of nutrient medium? As you can see in second line, I have underlined a nutrient medium is required for the growth and multiplication of an organism or plant part in culture. So nutrient medium help in the multiplication and growth of an organism or its plant part. What is the term used for the plant part for the culture? It is called as explant. When we have started this chapter, I have discussed the term explant. So next, third line, you can see the nutrient medium are divided into two main categories. Okay, there are two main categories. Fourth line, liquid medium and semi-solid medium. So what is a liquid medium? The liquid medium is prepared by dissolving appropriate proportion, or you can say percentage, of all required chemicals in distilled water. Remember always, if you are making a medium, then you have to use distilled water. So liquid medium is easy to prepare. Just we have to take the required chemicals for the preparation of medium and to dissolve in distilled water. But the percentage should be exact. Amount should be exact. Now, a semi-solid medium is prepared by adding agar into the liquid medium. As the name suggests, semi-solid means it will contains liquid as well as solid. And which solid you have to take for the culture? Agar. Do you remember from where agar is extracted? An algae is used for the extraction of agar agar. Remember? If anybody remember, then response me. Otherwise, see in your book of first year or from Google you can see the algae which is required for the extraction of agar. Okay. So you have underlined this and we have discussed about the nutrient medium. Now what are the different nutrient mediums uh, for this? We will discuss in two categories. First one is inorganic nutrient and second one is organic nutrient. Inorganic nutrient I have underlined, they are of two types, macro and micronutrients. As the name suggests, macro means which are required in larger quantities and micro means which are required in smaller quantities. But the important part for your competitive exams is their example. Macronutrients include carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, calcium, sulfur, magnesium. Okay, total 1, 2, 3, 4, 5, 6, 7, 8, 9. There are nine macronutrients: carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, calcium, sulfur, and magnesium. And the micronutrients are iron, zinc, manganese, cobalt, and molybdenum, including boron also. Boron, BO, boron. So the micronutrients are in less quantities, and it is 1, 2, 3, 4, 5, 6, 6, total 6: iron, zinc, manganese, cobalt, boron, and molybdenum. Next, organic nutrients. Organic nutrients include carbon source, vitamins, amino acid and amides, and growth regulators. The carbon source, the most common carbon source is sucrose. Sucrose is the most common source of carbon. Vitamins. Vitamins include thiamine, pyridoxine, and nicotinic acid, including inositol. So there are four vitamins which are helpful in composition of organic nutrients. Next is amino acids and amides. There are different amino acids used as a organic nutrient, but the main is glycine. Main is glycine. Glycine. I have underlined this. Growth regulator. We have discussed about the growth regulator in our previous lectures. There are different growth regulators and retarders: auxins, gibberellins, cytokinin, abscisic acid, ethylene. Now, in reference to growth regulators, I have underlined exogenous supply of growth regulator is needed. Exogenous supply means कि you have to give or apply regulators or growth regulators from outside. Okay, it is the exogenous supply of growth regulators. So the next part related with this is which are the different growth regulators. I have underlined the major groups of growth regulators are auxins. Different types of natural and synthetic auxins are present: indole 3 acetic acid, indole 3 butyric acid, naphthalene acetic acid, naphthoxy acetic acid, and 2,4-dichlorophenolic acid. Remember all these types of auxins. Next is gibberellins. They are mainly gibberellic acid. Cytokinin, it includes, I have underlined this, acetic acid, cytokinin, furfuryl aminopurine. We have discussed in our previous lecture. Zeatin, benzyl aminopurine, and isopentyl adenine. Abscisic acid, which is also called as stress hormone. Now, we have learned about the different types of growth regulators. Now, how they help? If you apply them in medium, how they help? I have underlined this. The auxins regulate callus growth by supporting cell division. and causing induction of rooting. You please remember rooting part because it will differentiate it from another hormone. Auxins regulate callus growth. We will discuss about callus culture in our videos also. What is callus? It is an unorganized mass of cell, unorganized. So auxin regulate callus growth by supporting cell division and induction of rooting. Okay. Cytokinin promotes cell division and initiation of shoot buds. So it is an important question. Both are related with cell division, but which induce roots and which induce shoots? Auxins induce roots, root formation, and cytokinin initiate shoot buds. Next, the gibberellins promote cell elongation. Elongation or cell maturation is promoted by gibberellin. Abscisic acid promotes regeneration of shoot buds. So you please remember which hormone is responsible for induction of roots, shoots, and regeneration of shoot buds. Next, the last part of this today's video is what is a basal medium? It will be an important question of 2 marks. I have underlined in the third line, it is a general medium containing inorganic salts of major and minor elements, vitamins, amino acid and sucrose in a proportionate amount, which is used for general in vitro culturing. Students, what is in vitro? You remember? If you remember, then write in your notebook while preparing this topic. In vitro means I am leaving dash. So, what is a basal medium? It will contains inorganic salts of major and minor elements. It will contain vitamin, amino acid and sucrose. So, such a nutrient medium is called as basal medium. This is an important part of biotechnology. Without a nutrient medium, the process of biotechnology cannot be initiated. Without the sterilization method, the process of biotechnology cannot be initiated. So, it completes all about the nutrient medium, and you please continue this by writing your notes. It will be helpful. Always prepare notes in point wise manner. If you will write notes in point wise manner, then it will be easy to remember. So thank you very much.